All right, folks, let's talk about some, or Chapter 10, controlling microbial growth. Again, we're talking about viruses a little bit in this chapter, so it's not just about growth, it's about controlling and maybe preventing contamination or preventing you from contracting things or transferring things. So it's not just, it's a little bit of a misnomer to say um, controlling growth because viruses aren't going to grow, but um, I think you get what I mean, right? So if we look at antimicrobial agents and the history of, we have to define a few things, like what are drugs. Um, Common lexicon, if you were just to say drugs, then maybe you think about illegal things, but drugs are any chemical that can affect physiology, right? So depending on how it's used, drugs can be any classification of things or any number of things. Chemotherapeutics, seeing chemo usually makes people think of cancer or chemotherapy. Um, any drug that acts against a disease is considered a chemotherapeutic, right? It is a chemical that offers a therapy. So even though we use that term chemo now for something different, it's still technically a larger term that means more things, right? And then antimicrobial agents, antimicrobials are not antibiotics necessarily. Antimicrobials control a lot of microbes. Antibiotics control specifically bacteria, right? So antimicrobials is again a larger class. When we're looking for the ideal antimicrobial, right? So this would be exactly what we would want. Something that's readily available, inexpensive, chemically stable. This is a problem with antibiotics, for instance. A lot of them in liquid form need to be refrigerated, or they break down quite easily. Easily administered, so um, pill form, shot form, right? It has to be easily taken. Um, Sometimes even the pills that we take for antibiotics, they're so big, people have problems swallowing those. Being non-toxic or non-allergenic, right? And again, that can be a problem with antibiotics. So a good number of, well, more people think they're allergic to antibiotics than really are. There's been studies recently that people that claim to be allergic to things like penicillin, for instance, are probably not actually allergic to it. Um, for instance, if you took a round of antibiotics and you say you got a little bit of a rash, um, that's not usually considered enough for you to say that you're allergic to it, right? If you're allergic to it, then you're going to have some serious reaction. Um, Again, that's not medical advice from me, but that's what the research says, is that it seems the majority of people that claim to be allergic to certain antibiotics aren't really, right? And then you want those antimicrobials to be selectively toxic. Again, that's a problem too, because living organisms are so closely related, it's hard to do that all the time. So it's one of the reasons that you might end up feeling kind of crappy if you're taking some drug to control some pathogen, is because it's also probably affecting some of your cells too, and that can make you, or in the case of antibiotics, it's affecting other microbes that are good for you, and then that can cause you to feel not well too. So being selective is a issue. And on the issue of selectivity, one of the things we could use for selectivity would be viruses. Viruses are pretty selective in what they infect, and so if we could use a virus to infect only the bacteria that are causing us problems, that would be good to use viruses as an antibiotic of sorts. But again, the problem is trying to convince the public that we should release a modified virus into them to help control an infection. Not an easy sell there. Some folks through history, so Paul Ehrlich, he was looking for these kind of magic bullets, so things that could control, um, well, microbes, right? And so he was one of the first ones to find these arsenic compounds that killed microbes. If you know anything about arsenic, it's not particularly good for humans. It will control microbes, there's no doubt about it, but it's also highly toxic to you. Fleming um, was the first to notice that penicillin was released from the fungi Penicillium, and I'll show you a slide on that next. It was kind of accidental. It's what happens when you get a little contamination in the lab, but you also make really good observations. Domac was the first one that discovered the sulfonamides as an antimicrobial agent. And then Waxman noticed that other organisms besides penicillium also make antimicrobial agents. And typically in the microbe world, antimicrobial agents are used by the organism to help um, get rid of other organisms, right? So if I just go to the next slide, so this is the penicillium fungus, right? That's releasing penicillin. And so what that does is it kills the bacteria around so that any food source that may be there is still available to the fungus. Um, Bacteria themselves can make their own types of antibiotics or antimicrobials. What that would do is it would restrict the number of microbes that could grow close to them, therefore, they can better utilize resources. And so you end up with this kind of back and forth war between the two. So once certain bacteria are then resistant to penicillin, then they can invade the penicillium space, and so you get this back and forth kind of coevolution thing happening, right? But that's why other microbes, right? Because you think, well, if antibiotics kill bacteria, why would a bacterium be making an antibiotic? Well, it's because they're selective and it only kills certain bacteria and certainly not all of them. When we look at antimicrobials, they can either be semi-synthetic or synthetic. So semi-synthetics are antibiotics that we may have found naturally and then just altered them a little bit to make them maybe longer lasting or more effective or maybe more stable, right? So that they don't degrade quite so quickly. And then, of course, we can make fully synthetics. All you need to know is if you're trying to make an antibiotic that is a protein, well, all you need to know is the DNA, and you can synthesize those pretty easily in the lab. So this is just showing you organisms that we got individual antimicrobials from, right? And they did all originate from some organism, right? And this is kind of what I'm getting at with that coevolution thing, right? So you have this constant war between organisms, and then we as humans have just exploited that for our benefit, right? And so it helps us. Although what we see is that that war continues, right? So, for instance, we use penicillin, right? We overuse it, use it incorrectly, which encourages, I shouldn't say that, it selects for bacteria that are resistant to penicillin, and so we just end up with, again, resistant bacteria. If you leave my course not knowing anything but this, um, I'll be happy, right? So even at the onset of this COVID thing, people were talking about um, using antibiotics, right? That doesn't work. It doesn't work that way, right? Like antibiotics don't kill viruses. You need antivirals to kill viruses, or you need something that maybe maybe an alcohol concentration or whatever that just denatures proteins, causes harm to DNA, lipids, whatever, right? Um, Antibiotics are effective against bacteria only. They are not effective against flu. They're not effective against COVID. They're not effective against any virus, right? Antibiotics only work against bacteria. Please know that, right? So mechanisms of action, again, we want them to be selectively toxic. Um, and again, that can be a problem, especially when you start treating eukaryotic infections. What are eukaryotic infections? Well, most protists, like amoebic dysentery or giardic dysentery, um, those are eukaryotic organisms that are causing you problems. And so finding a drug that is selectively toxic to them and not toxic to you can be a problem, right? It's also the issue with antivirals, because viruses tend to be a huge component of a virus is either DNA or RNA, and because they're not living, so you can't really, I mean, you're not gonna attack the mitochondria of a virus because it's not there. You're not gonna attack some metabolic pathway for a virus because it's not there, right? And so attacking viruses can be a problem that way too. If you try to interact with their DNA or RNA, well, you have DNA and RNA too, and so that can be a problem for you. Right? So this just shows some of the ways in which we can try to stop infections or stop microbes from causing problems, right? So we can inhibit cell wall synthesis, we can inhibit protein synthesis, we can disrupt cytoplasmic membranes, we can inhibit metabolic pathways, inhibit DNA or RNA synthesis, or we can inhibit the attachment of the organism to the host cell. And one thing I just said there reminded me of something. And hopefully you guys know this by now. I saw this thing going around Facebook yesterday about uh, this doctor, and I won't go into too many details of why I'm air quoting doctor. He was telling people to take a certain homeopathic medication, which I think that is an oxymoron. Um, he was telling people to take that in order to make their cell walls more permeable, right? You don't have cell walls. So when you hear a doctor say something like that, you should be highly suspicious of them. Um, and it turns out that this individual was trying to sell some stuff. Shocking, right? Um, but it's just something that you should be aware of when you hear people say, because the general public would say, well, David, you're nitpicking, right? Well, if it's a cell wall or a cell membrane. And maybe if that's Joe Schmo on the street, but if this guy's claiming to be a doctor and he's talking about humans having cell walls, that's a red flag, right? So ways we can inhibit cell wall synthesis in bacteria are through beta-lactams. Beta-lactams are essentially penicillins. Um, they cross-link, right, and I can show you here. So if you remember, in the cell wall of bacteria, you had these NAG and NAM molecules that built that peptidoglycan layer. Well, if you can either stop new NAG and NAM molecules, or if you can prevent cross-linkage, um, then you can essentially weaken bacteria, right? And that's what these beta-lactams do. They essentially come in here and they bind. So think about taking a chain link fence and you can't slide it across each other anymore. You bind it in place. And so that if you try to add new molecules in there, it makes the cell wall get all kinked and bound up on itself, and essentially causes it to poke holes in itself, right? That's how these penicillins work, right? They cause that um, osmotic pressure essentially to burst the cell because you weaken um, the peptidoglycan layer or the cell wall. Right? Now, one of the things that happens here is some of these bacteria can produce, if they're resistant to penicillin, what they can do is come in and they'll degrade this. Right? They have an enzyme that breaks this down, and that's what allows them to persist in those environments. Right? So when we're looking at the semi-synthetic derivatives of beta-lactams, one of the things that we've been able to do is make them more stable in acidic environments. Why? Well, your stomach is super acidic. If you take an antibiotic and it ends up in your stomach for a period of time, it better be effective once it leaves there. Otherwise, you've taken it for no reason. Make it readily absorbed. If it can't leave your small intestines, it does you no good unless the infection is right there in your small intestines. How susceptible it is to deactivation, which means how susceptible is it to bacteria breaking it down and making it not function. Right? And then how active is it against other types of bacteria? So does it only affect one or does it affect multiples? Sometimes you want to take an antibiotic that's effective against multiple bacteria. Um, Not always, but sometimes that can be effective, right? And so that's one of the one of the things that we can synthesize um, in the semisynthetics, for instance, to make them better at doing these things. Let's see, I just talked about that, right? So as far as increasing the amount of peptidoglycan, if you can stop that, you can stop bacteria from growing. Fungal cell walls are a little harder. They don't look exactly like bacterial cell walls, so you have to use something different, right? So um, fungal cell walls have this glucan molecule in them. I won't get too much into the weeds there. Um, but you have to use these echinocandins in order to inhibit those glucans in order to stop fungal infections. Fungal infections are usually harder to control, again, because they're um, eukaryotic, so they can be harder to control than prokaryotic infections. But there are plenty of drugs to do that now. In protein synthesis, you can target the ribosome, right? Prokaryotes have a different ribosome than eukaryotes. The problem there is mitochondria in eukaryotes have 70S ribosomes just like prokaryotes do. So if you're taking an antibiotic that is attacking, quote unquote, the 70S ribosome in a bacterium, um, then it could also affect your mitochondria, and that might be one of the reasons that you feel lousy while you're taking that antibiotic, is because it's, again, mitochondria make ATP. If you affect them, you're not going to make as much ATP. You're going to be lethargic. You're going to have, you're just generally not going to feel well, right? So protein synthesis. If you can interfere with the RNA molecules, specifically here transfer RNA molecules. Here they say charging. So maybe you remember, let me increase this. You might remember when we talked about RNA molecules. Let me change colors here. The transfer RNA molecules were the ones that look like a T, right? And then they had the amino acid up top, right? So if this one happens to be this particular amino acyl transfer RNA, then that's the particular, um, like that might be the particular amino acid, or it might be isoleucine or whatever, right? So if I was to say iso, right? Well, that tRNA, once it drops that amino acid off, right? So it'll just look like that without the amino acid stuck on top. Um, if you can prevent the charging, so when I say charging, what I mean is, so in the case of isoleucine, if this is the tRNA molecule that transfers isoleucine, this would be the tRNA in an uncharged state, right? It's charged when you actually put the isoleucine on it, right? So if you can prevent that charging, you can prevent uh, the use of that particular amino acid in making a protein, and so you might could prevent um, making proteins in general. And if those are important proteins for a molecule or for a organism to live, then you've prevented that by using certain antimicrobials, right? So prevent loading there. Let's see, cytoplasmic membranes. Usually what you're trying to do in cytoplasmic membranes is just punch holes, right? So rather than go through all of this, although I would point out here that so these particular ones, um, so the amphotericin Bs, the nystatins, or nystatins, so that statin, you might have heard that term before. The problem with these is that they affect cholesterols. Cholesterols are a normal part of a cell membrane. Um, but if you affect them too much, then what can start happening? So if this is the amphotericin B, what it does is it'll bind essentially to the ergosterols, and it causes these holes to stay open and form inside the cell membrane. And so you get contents moving in and out pretty easily, and that can cause the cells to rupture, right? So a lot of water can influx in, for instance. The problem is cholesterols, right, are a normal, again, they're a normal part of cell biology. And so if you affect cholesterols in human cells, then that can be a problem too. So uh, again, you can control this through dosage, right? So if you're not taking a whole lot of it or overtaking it, then probably not as big a deal, right? But it certainly can be. Right? Anti-metabolic agents. So if you can stop an organism from metabolizing, obviously you can stop it from functioning. There's a lot of different ones of these. Again, remember, this is one of the reasons that we talked about things like electron transport, right? So if you can interfere with electron transport, you're probably going to kill an organism because it's not going to be able to make its ATP, right? Certain heavy metals will inactivate a lot of enzymes that are needed for uh, just normal function inside of an organism. If you can stop glucose uptake, right? Glucose is the source of energy for most living organisms, right? So if you can stop that, then you can essentially starve an organism uh, and kill it, right? Um, if you can block the activation of viruses, so whether that be them attaching onto cells or injecting DNA or an RNA into a cell, plenty of ways to do that. But that I don't really consider that a metabolic pathway. Virologists would disagree with me though. And then if you have, so here they're just saying this is kind of a dustbin here, any metabolic antagonist, right? So an antagonist is just anything that is counter to. So anything that would be counter to metabolic activity, obviously, would be a way to interfere or inhibit those pathways and stop microbes from functioning. Right? Nucleic acid synthesis, if you can inhibit that, typically we only see this in humans if you're going to treat things like cancers, because you don't want to inhibit nucleic acid synthesis normally. Um, it's just not a good idea in normally dividing cells, right? Chemotherapies will do that. They will stop nucleic acid synthesis. But that's also why you get the range of symptoms that you get with chemos, right? So you end up with gastrointestinal issues, hair loss, skin conditions, maybe sores in your mouth, right? Those are all epithelial tissues, and epithelial tissues rapidly divide, and so they need to make a lot of DNA. And so if you stop that through the use of chemos, that's what causes um, a lot of the side effects that you have there. The plus side to the chemo is cancer cells can't repair themselves like normal cells can. So if you damage the cancer cell, the hope is that the chemo will kill all those cancer cells before it does so much damage to the rest of the normal cells. Um, that's the hope, right, with chemos. Right. Let me go back here so I can kind of tell where I'm at. Okay. Let's see, where was I? Okay. Um, so we have these things called quinolones or fluoroquinolones. Um, remember when we talked about DNA synthesis, we talked about an enzyme called gyrase. Gyrase prevents the supercoiling of the DNA molecule. Well, if you can act against that, then the DNA molecule will supercoil and it'll shear off, break itself, and that will stop it from doing what it normally does. The other things you can have are these analogs, right? Nucleotide or nucleoside analogs. So an analog is something that just looks similar. And so we have all of these, right? All of these drugs are nucleoside or nucleotide analogs. So what that means is maybe in place of a thiamine, right, you end up putting something else there, right? Or instead of an adenine, you end up putting something else there in the DNA molecule. And if you do that, then you're going to stop that DNA molecule from being able to synthesize itself correctly. Um, the problem with these drugs, though, is they can often be cancer causing, right? Because if you put I mean, we talked about that in that chapter when we were talking about mutations, right? And so if you use some analog, then that can certainly cause mutations, and mutations can certainly lead to cancers, right? So that's one of the problems with nucleotide analogs. Inhibit RNA polymerase, right? So remember, you need RNA polymerase to make a lot of things. Ultimately, you need RNA polymerase to make RNA, so you can make messenger RNA, so that you could make a protein. In the case of viruses, so again, remember that when we talked about replication, we talked about, I don't know why it always defaults to red, and I just don't like red. I mean, I like red to wear, I don't like red to write with. But remember, we go from DNA to RNA to amino acids, right? That's the typical pathway. However, smart as they may be, some viruses, they're called retroviruses. Um, the viruses, so inside them, right? So if this is the capsid of the virus, so in here would be RNA and not DNA. And so what they do is they inject that RNA into your cell, and inside that RNA they also have what's called a reverse transcriptase enzyme. And so what that reverse transcriptase does is it changes RNA back to DNA. And so now, once that viral RNA is changed to DNA inside of your cells, right, that DNA then goes through DNA to RNA to protein, and so that you're essentially making more viruses. So if you can inhibit reverse transcriptase, then that's one way to control um, viruses, right, through that enzyme. Humans don't normally have reverse transcriptase in them, right? And so that's an easier one to target as far as viruses go. So here's a question. Some antimicrobial drugs are harmful to humans, so why can physicians safely prescribe such drugs despite potential danger? And this goes back to one thing that I constantly preach, is that the dose defines the toxin. So anything in a high enough dosage is problematic, right? So too much water is a problem, but water in the correct dosage is good and needed, really. Right? So even though certain drugs are harmful to humans, if you take them correctly in the correct dosage, then they're not. And again, that's with anything, right? And when we talk about antibiotics, or I guess I could more generally say antimicrobials, we can also talk about them as being narrow or broad spectrum. Typically, you want to use the narrowest spectrum antimicrobial that is also still effective. Um, what that does is it helps reduce the chance of resistance from some other organism. So, for instance, if I use bacteria as an example, if I had some, I don't know, some infection, right? So I had a staph infection. If I could take an antibiotic that only harmed staph bacteria, then that would reduce the chance of me having some other infection occur. Um, when you start using broad spectrum antibiotics, though, you do increase the chance of getting these super infections. Why? Well, because the numbers just increase, right? So you're trying to control more bacteria, and so if you're trying to control, here when I say more, I mean more species. So if you're trying to control more species, the numbers might be more. The chance, I mean, you're just gambling at that point, right? The chance of there being a bacterium that is resistant to uh, that particular broad spectrum antibiotic is increased, and so that can be a problem. The other problem with broad spectrums is it'll certainly kill more of your normal flora. Um, I have a problem with that term. Flora typically means, so you have flora and fauna. Flora typically means plant life. I think the correct term here would be fauna, but I digress. Anyway, broad spectrum antimicrobials or antibiotics would kill more of your normal bacteria, and again, that can make you feel quite lousy, right? Um, and again, taking these incorrectly, I mean, that's C. diff. That's what that is, right? That's the way that you get C. diff, is to take antibiotics incorrectly. And anybody that knows anybody or has interacted with anybody or has had it themselves, you don't want that, right? That's a bad day for sure, right? And so this shows you some of the spectrum of certain antimicrobials, and so you could see prokaryotes, eukaryotes. So notice some of these, right? Some of these sulfonamides um, are effective against some eukaryotes, right? Like some protists, for instance. Notice the erythromycins, pretty effective against a lot of bacteria. Penicillins, mostly gram positives, but maybe some gram negatives. These polymyxins are gram negatives only. Streptomycins, the mycobacterium can be hard to control because they have a really like waxy layer on them. Um, but gram negatives too, right? But what you would want to do here is, and this is why they're trying to culture maybe, so if you get strep, right, they want to culture that to make sure it is what it is, so um, they can kind of select for. But this is also why, for instance, maybe if you have strep, they just give you erythromycin because, I mean, it can control a lot of things. Um, but again, using a broad spectrum is not always the best idea, right? You want to be as defined as you can, um, if you can. Right? And so how do we know the effectiveness of these antimicrobials? Well, there's several ways we can kind of figure that out. One can be the diffusion susceptibility test. So this is something that we would have done in lab. Um, minimum inhibitory concentrations or bactericidal concentrations. So for instance, this diffusion test, right, this is what we would have done in lab. You take a plate, you grow bacteria on that plate, or you take bacteria and you spread it all over the plate. And then after you spread it, you put these discs on the plate. And these discs are impregnated with an antibiotic. Right? And so what you're looking for is, do you get a zone of inhibition around that antibiotic? Right? So what this means is, so from, let's see, right? So from here to here, right? There's no bacteria growing there, right? Why? Because whatever this antibiotic is prevented it, right? But if you notice here, I think the G is gentamicin. The bacteria grow right up to the edge of that gentamicin disc. So gentamicin has no effect over whatever this particular bacterium is. It's probably E. coli. I don't know. The other thing you're looking for is, what's the distance of this zone of inhibition? If it's pretty close, right? So notice, I think S is streptomycin. So notice this one is less, right, than this one. It doesn't mean that you necessarily want to use this antibiotic, right? This one might be just as effective, but you also want to measure this because with every one of these discs, you need to know what bacteria you're working with, and they'll come with measurements, and so you'll measure this in millimeters, and it'll tell you, is this bacteria um, resistant to, or susceptible to that antibiotic, or you might get a measurement that's kind of in between those, and so it's kind of hard to tell. So what you would want is probably something that's not too broad, right? Because that would mean a broad spectrum antibiotic that maybe you don't need that strength, that level of, but you don't want something that's too close either, because then it might not be as effective, right? But that's one of the ways you can look at those. Another way you can look at them, let me show you this one first. So what this is doing is looking at individual antibiotics at a specific strength, right? So for instance, I can't remember the units they use, but the 30, so I think this is tetracycline, and the 30 indicates the concentration of, right? So the 0.25 30. Most of these are 30. There's a 10. Right, so that indicates the concentration or how strong the dose is. The other thing you can do is this way. So this is on this strip, it's one antibiotic, and the dosage or the concentration varies from the end of the strip up, right? And so what you're looking for is at what concentration does it start to inhibit the bacteria on the plate, right? And so you wouldn't, you wouldn't want to give a patient the absolute strongest concentration possible, right? That's going to cause them some negative side effects. So what you want is some Goldilocks concentration here that works best. Right? The other way to do minimum inhibitory concentrations is to use these wells, right? So what you have down here at the bottom is increasing concentration of the drug, um, and then you can look at how turbid the wells are, right? So turbid meaning how cloudy they are. And so the less cloudy they are, the more effective the drug has been, right? So these darker green ones over here, the drug's not effective at all. And so notice as it starts to pale out a little bit, so maybe the concentration you want to use is somewhere in here, right? And certainly not past that necessarily. Here they're talking about, again, it's just another way to figure out minimum bactericidal concentrations. So they're just varying the concentration of the drug, right? So 8 micrograms per milliliter or 16. And so here it's showing you that 8 micrograms is not effective, 16 micrograms is. And if 16 micrograms is, then probably 25 is too. But this would be probably the concentration that you would want to use to control that bacterium, and maybe not more. Right? So plenty of ways to deduce those things. Um, Certainly plenty of routes of administration for these. You've probably experienced all of these. Maybe you've not. Maybe you haven't had some intravenous. I've certainly not. Well, I have. I was given IV antibiotics whenever they thought I had appendicitis one time. But it can certainly be topical, right? If you use something like Neosporin, right? That's topical. It could be oral, right? So you're taking a pill or a liquid. Intramuscular, right? So a shot. Intravenous, obviously through your veins. That's So I think I did when I was much younger. I was in middle school, and for a couple days I just had severe lower right abdominal pain. Um, wasn't hungry. I never had a fever, right? But I wasn't hungry. Just didn't feel good at all. Ended up in the hospital because they thought I might have had appendicitis, and this was so again back when I was in middle school. They didn't. I think now they can do maybe ultrasounds or some kind of scan in order to actually see your appendix to see if it's inflamed or not. They didn't do that back then, and so essentially it was just a wait and see for me. They put me on IV antibiotics, and after a day or two, my symptoms went away. And so that seems to be a more common way um, if you can catch it early on. Then it seems like they're actually doing that now for appendicitis more so than just the common just go in and remove it type of thing. But again, it depends on how severe it is. Right. And so this just shows you relative concentrations of drugs uh, in tissues and the administration, right? So oral administration, obviously it increases slowly and then drops off slowly. Intramuscular, the increase is rapid, the fall off is rapid, and then of course with IV, it the increase is rapid and it just maintains that for long periods of time, right? This could be again they're talking hours. This could be days. It just depends on how long the administration is. Right? Although I will say it's so I got a poison ivy, and so I had poison ivy one time that then got infected, and I was put on antibiotics. I took one dose of the antibiotic that night, and then by the next morning, it was just unreal how much better I felt after that. which begs the point you shouldn't stop taking them then, right? If I would have stopped taking them then, I did feel so much better. But if I would have stopped taking them then, any of the bacteria that would have been resistant at that point, man, they would have just flourished, and that would not have been a good time for me at all, right? Again, toxicity is an issue. You're looking at the therapeutic index, so you want to know what's the most effective concentration, um, what's the least amount I can take, and it still work, right? The other thing you want to pay attention to, obviously, is pregnancy. If you're interested in going to look at how drugs can affect pregnancy, go Google flipper limb syndrome, right? Flipper, Flipper. So flipper limb syndrome was caused by thalidomide. Thalidomide was a drug that seemed to work really well against nausea, for instance. And so, what individuals are consistently nauseated? Pregnant women, right? They have a lot of morning sickness. The problem with thalidomide, and I'm not going to talk about chirality necessarily, but the chirality of a drug is the way it looks in 3 dimension, right? And so when you look at it in 3 dimension, well, it's kind of like looking at your hands, right? And so I'll do that instead of draw, right? I can't, right? These structures look the same, right? My hands do. I can't take this hand and put it over this one. It doesn't work that way, right? I have to put them facing each other as if they're facing a mirror, right? So in that sense, they're not the same, right? Gruesomely, if I was to cut my right and left hand off and put them somewhere, you would know which was the left hand and which was the right hand, right? Just by the orientation, right? So they're different, even though they look very similar. That's the same thing with drugs. And with drugs, we can talk about chirality, right? Whether they are essentially drugs that are counterclockwise, right, or clockwise drugs. Um, and again, that just has to do with the orientation of the three-dimensional structure. But what we found out with thalidomide is this mattered so much that, and I can't remember if it was the left or the right hand. Um, the drug was the same. Thalidomide was the thalidomide. It had the same number of the exact same atoms. It's just one was counterclockwise, chiral, versus clockwise. That 3D shape alone was enough to cause flipper limb syndrome, right? And so, in that sense, knowing whether or not uh, pregnant women should take it, I mean, it's not going to affect men, right? Men, in fact, one of the problems with thalidomide, it worked so well that if men were going to take it, they started asking men if there were women in their lives that were, um, or that could give birth, and if so, they a lot of times they wouldn't give it because the temptation for women to take it, it worked so well, the temptation for women to take it was too high. Um, this went on a lot in other countries. In the United States, it didn't go on so much because it was regulated so heavily. You can still get thalidomide though, but it is one of the most heavy regulated um, pharmaceuticals out there. Again, because it can cause such devastating um, birth defects. Um, I think it's given a lot though in cancer patients, right? So if you're getting chemo, um, pregnancy is probably not something, or getting pregnant is something you probably shouldn't consider uh, because of the way it works, right? But thalidomide, if you've not heard about that, or flipper limb syndrome, that's an interesting bit of history there that you should look up. It's quite interesting. Side effects, I talked about some of these already, right? So allergies, of course, we should be aware of those. Certainly to the point of anaphylactic shock, right? That's a huge problem. And then what does it do to the normal microbiota, right? That can cause you all sorts of problems if you affect your normal microbes, right? Microbial resistance, I won't read through all of these, but microbes can be resistant in a number of ways. So for instance, they can produce biofilms that prevent the antimicrobials from penetrating. They can have mutations that cause them to be um, resistant to, right? Here they're showing you how certain enzymes, right? So there's an enzyme called penicillinase that breaks down the penicillin, right? And so if a bacterium makes that penicillinase, it effectively inhibits penicillin from functioning correctly, right? Penicillinase is an enzyme. So if it's an enzyme, it's encoded in the DNA, and if it's encoded in the DNA, that DNA can be shared between bacteria, right? Could be on a plasmid, for instance, right? So, but a number of ways that bacteria can have some resistance there. They can develop multiple resistance, right? And that can be a huge problem, or even cross resistance. Um, so if a pathogen has that resistance plasmid in it, and it's exchanged, so if I already have one microbe that already has a resistance plasmid and it picks up another one, now it's multiple resistant. We tend to call those super bugs, right? Multiple drug resistance pathogens, right? Those are the MRSAs essentially of the world. Right? The way to kind of retard that resistance is to, well, if you're talking about on surfaces, the way to retard that is to use different things over time. The way to retard that resistance in situ or in tissues is one, um, make sure you're using the correct antibiotic for the correct infection. Make sure you're using the correct dose. Make sure you're taking all of it, right? That'll help reduce that. Sometimes you might take multiple antibiotics that act in synergy to each other, right? And what do I mean by that? Well, if I show you, so this is one antibiotic and another. And so what you can see here is that this particular antibiotic by itself gives this much of a zone of inhibition, right? This one by itself gives this much of a zone of inhibition. When you put them together, you get more, right? So the zone of inhibition increases greatly, right? And so in that sense, if you use those two together, it gives you a synergistic effect. And so the two of them together work better than they do singly. Sometimes, though, again, that's very broad action. It's probably not your first line of defense there. You probably want to explore that later because you would increase the chance, or you could increase the chance of selecting for things that are multiple resistant even. So, I mean, there's this constant kind of teeter-totter, right? You certainly want to make sure that there's no antagonistic results between drugs. Otherwise, you're really kind of shooting yourself in the foot there, right? Um, and then, so what we have seen now, and you may have heard your doctor talk about this too, is second and third generation drugs. So second and third generation penicillins. Now, with penicillin, for instance, we're so far down the line, second, third, fourth generations, that what we're finding is that if we go back and use some of the first or second generation medications, they're almost more effective than some of the newer ones. And so it may be that some of the old antibiotics start working well again because we haven't used them in such a long time. But that's another way to help retard that resistance is to kind of re-engineer some of the antibiotics that we have and hopes that they'll work better. And again, maybe that opens up the door for us to use some of the older ones. Right. See, I forgot what this question is. Why is it incorrect to say that individual bacterium develops resistance? Oh, so the reason this is incorrect is, so if you expose a bacteria to an antibiotic and it's not already resistant to that antibiotic, it's going to die. I mean, there's no way around that. So the antibiotic does not make the bacteria resistant. All it does is selects for those that already have resistance. That's what the antibiotic is doing. So if you have a billion bacteria, right, and you take an antibiotic and almost all of those bacteria are susceptible to that antibiotic, they're going to die, except for the few that aren't. And then your immune system hopefully will take care of those. And then you keep taking the antibiotic, your immune system, along with the antibiotic, helps control the infection. If you stop, though, you stop taking the antibiotic, then all those resistant ones, well, the other thing I should say is just because an antibiotic doesn't kill a bacterium doesn't mean it's not controlling it. So a bacterium can be resistant to an antibiotic, but it still might not like it very much in the sense that bacteria can like things. So maybe the bacterium just doesn't grow very well in the presence of the antibiotic, even though it doesn't die, right? And so if you constantly expose it to it over 10 days, eventually it can kill it along with your immune system. But if you stop at day 7, right, then those bacteria that are still there, right, they just weren't very happy. Now they're released from that pressure and they grow like crazy, and then you've got a serious infection that doesn't respond to those antibiotics, right? And so that's probably a good place to stop with chapter 10 right now, and then I'll post the rest of it a little bit later. So first half of chapter 10. Again, if you have any questions, let me know. Happy to answer.